a Princeton, poi è stata postdoc a Stanford. Come dicevo, nel 93 è diventata professoressa a Caltech, questa prestigiosa università della California. È diventata anche, dove è professoressa di neurobiology professoressa di neurobiologia. È diventata Our Use Investigator in quegli anni. E adesso si è un po' annoiata dell'America, infatti ci farà vedere delle diapositive un po' critiche, e ha deciso di venire in Europa e ha preso una posizione come director del Max Planck, che verrà costruito a Francoforte, e quindi nei prossimi anni lavorerà in Europa, quindi abbastanza vicino a noi. Quindi ringrazio moltissimo la professoressa Schumann per aver accettato il nostro invito e quindi la prego di...
Thank you very much for that introduction, Antonio. Okay, so what I would like to do in this discussion is to, again, talk about some of the topics that Professor Malgoroli introduced, having to do with the brain, its structure, the synapses, and discuss some of the ways that we know that synapses can store information, and how scientists study learning and memory in the laboratory, and then finish with a series of what I think are very exciting studies that we've done in human subjects, where we have this unique opportunity to actually record from human brains while people are learning and remembering. So perhaps the most simple question that is where in the body are the things encoded and stored when we learn and remember something? So of course it's the brain, and here is a picture of the human brain, and you can see that the brain is actually not smooth, that it's folded, and the folding of the brain serves the purpose of endowing us with an even greater intellectual capacity, because it's like taking a sheet of paper, essentially, and crumpling it up so that it can fit into the skull. So the folds that you see here actually increase the real estate, or the number of synapses, number of brain cells that we can fit into our skull. And I show here, just out of interest, the brains of other animals, the elephant, the dolphin, the gorilla. And as you can see, the size of the brain roughly correlates with the intelligence, and you can also see that there's a decrease in the complexity of the brain, fewer numbers of folds. And you get down here to the lowly mouse brain that has hardly any folds at all, and shows us that the mouse is actually not very smart. So if you look in the brain and you cut, say, a section through the brain, you see a beautiful organization of the cells. So these are the principal cells, those are the neurons, and here you can see a sort of stripe that represents where the cell bodies are here, and these processes extending here are the dendrites that Professor Malgoroli referred to. So as he explained, every neuron has basically two compartments. Well, let's say three. It has a receiving set of processes called the dendrites, and it has a set of branches that send information to the next cell in the line. And this is called the axon. So the axon of one cell comes in contact with the dendrite of another cell, and at this specialized point of contact is the synapse. So here I've just shown it again, the terminal of the axon that Antonio showed you, that this is where the neurotransmitter is packaged in vesicles, comes in close contact with a specialized compartment in the dendrite, and that's the synapse. Here's an electron micrograph of a synapse, and this pink compartment here is the presynaptic nerve terminal, and you can see these are the vesicles. These are about 50 nanometers in diameter, and inside these vesicles are the individual neurotransmitter molecules. And you can see that in this cross-section, so this is again a slice, that the presynaptic axon comes in very close contact to the postsynaptic, so that's the receiving part, the dendrite of this cell. And right in this gap here, which is called the synaptic cleft, this is where the neurotransmitter diffuses across to interact with specialized receptors that are present in this little black area here. Now, in that black area are all of the proteins that are packed in that we spend a lot of time studying as neurobiologists. So there are the proteins that are the receptors for the transmitter, as Antonio described, the glutamate receptors. And then there are also many other proteins that are essential for the functioning of the synapse. And the ability of this particular synapse to respond to transmitter depends on not only the receptors that are there, but how many receptors are there, what their state is, and also how they're clustered. So depending on where the transmitter is released, the clustering and the presence of the receptors in the membrane is very important. And many of the drugs that we hear about and we read in the press that are important for neuropsychiatric disorders and neurological disorders, like, for example, Prozac, are drugs that target the synapse here and that change the availability or increase or decrease the efficacy of the transmitter at the synapse. So one of the most important features of brains is their ability to change. So the goal of our brains and other animal brains is to perceive the environment and react to it. And we, as animals with nervous systems, we need to be able to learn because we need to learn from experience. And so in order to learn, then our synapses need to learn. So they need to be what we call plastic, or the ability to, plasticity is the ability to change. And synapses are plastic. And of course, one way that memories are stored is by changing the strength of communication across synapses. So how can we actually study synaptic plasticity in the lab? So Antonio described some experiments that I will just describe a little in greater detail here. So one thing we can do is to have access to synapses, and we can take an electrode, a glass electrode filled with the salty solution, and we can actually take that electrode and impale one cell so that we can record its electrical activity. And then if we know the cell that's talking to that cell, say this cell here, we can stimulate this cell and record the response. So we can measure the synaptic communication between these two cells. And what we can do is measure, as Antonio showed you, some of the actual electrophysiological traces that we can record. So we can measure how big those are. And in a typical experiment, we would stimulate cell 1 and record from cell 2 and measure the size of the response. And over time, if you repeatedly stimulate, most synapses are fairly reliable and constant. So you will always record a response of roughly the same size. But what you can do is actually mimic what might happen in the environment when we encounter a new stimulus or we learn a new association. And the way that we do that is to rapidly stimulate one cell. So that's shown by this arrow here. And what we have observed is after that rapid electrical stimulation, now the synapse has changed. So now the synapse is increased, it's enhanced. And what's interesting is that enhancement can last a very long time. And in small animals and in rodents where we study it in the lab, it can last for up to months. So this observation that was originally described by Bliss and Lomo is exactly the kind of feature that we would look for if we looked for a substrate in the brain for storing information. So now what a lot of scientists do is try and understand the mechanisms that allow the synapse to change, not only the mechanisms responsible for initiating the change, but what is it that allows the synapse to stay elevated for a long period of time. Now, our brains are made of proteins, and proteins have a half-life. So proteins get made and they get turned over. And the range of half-lives of proteins is between about 20 minutes and a month.